Welcome back to the Hackers Notepads. My name is Christian Servin, and today we're gonna have a great plan for a great show. Actually, it's titled Synchrotron, Nanotechnology and Data Science by Hiram Castillo-Michel. Just want to remind you about this podcast that we actually started. We built it up just to increase the interest in computing, general computing. It doesn't have to be in computer science or information technology. If you have noticed, we have done a lot of different episodes that will basically impact computer science in the aspects of humanities, social sciences. And today we're going to talk about something that impacts in chemistry and in particular in environmental aspects. Let me just talk about real quick about Dr. Hiram Castillo Michel, who is my friend for many years now, right? I met him when we were at, well, probably I'm not so sure we were at UTEP, but we have overlap in many different areas, in many different stages of our lives. But Hiram, he is a UTEP, University of Texas in El Paso graduate. So he graduated from, and he will tell you a little bit more on his slides. It's a great slideshow that he has provided. But he graduated from UTEP here in El Paso, Texas, but he has been working in many different national institutes and national labs across the country in the United States. And now he is in Grenoble, France. So he's been there for a couple of years now, working in Synchrotron, which is one of the, I think it's one of the largest synchrotrons in the world. And Hiram, he belongs to the community. For many years, he was here in El Paso, part of Mexico. He goes back and forward. He has a lot of collaborations in different states in Mexico and also in the United States. You will see all the work that he's been working with Berkeley, universities, and whatnot. Hiram, he will give us a very brief description about the work that he's been done for the past years. But I just want to let you know that for every slide that you will see in Hiram's presentation, we can spend hours per slide, hours, because every single piece, every single bullet that he just created there, there's a big story behind that thing. So one of the things that I admire about Hiram is that he is super intense in the aspect of the chemistry aspect when you deploy it, the statistics behind that scenes, all the mathematical behavior that happens. And nowadays he's doing a lot of data science indirectly. He told me, you know what, I am not a data scientist, but I'm an expert in these areas. And now the beautiful thing that in the institution that he's working is that you have this collaboration among scientists, mathematicians, statisticians, computer scientists, chemists, environmental scientists. So this is the beauty. This is the whole beauty of having these collaborate aspects between scientists. So you will see in this presentation a couple of different perspectives that science has impact in real world problems. Notice that the work that he does is completely applicable. So this is real applications. Nothing wrong with theory, by the way, but I'm more like a theory guy, which basically we design algorithms and whatnot. But at the same time, when we put it in practice, you will realize that there's not many things that make sense. And you probably require a little bit more of integration of different areas in order to make things sense. And this is what Hiram will give us this explanation. And I know Hiram for many years, as I mentioned, and every time I have this conversation with him, we always spend hours. I remember when I saw him one day, Hiram, was this in an airport, right? So we spent hours there. So it was like, usually in the airport, you have this connection between flights, and it's like, okay, what are we gonna do with these things? It just went like 5 minutes, those hour and something, hour and a half, I cannot remember. So it's very interesting to see, and I'm very happy to see you here, Hiram. Thank you so much for joining us. Hiram, you wanna share something with us before we start the conversation?
Well, no, thanks a lot for the introduction, Christian. Yeah, so I agree with you that it's always a pleasure to talk to you. Yeah, all these kind things that you say about the work I do. I mean, the collaborations which I am involved, and I hope that I'm looking forward for this discussion we will have, this podcast. And I hope that you enjoy what I am going to be talking about. Just feel free to ask any questions, to let me know if something's not clear. And so I have prepared a set of slides, but at any moment we can stop and maybe if it's necessary deviate a bit or just go more in details, etc. So thanks. I am very happy to be today talking to you. And if you want, we can start if there's no more.
Yeah, we can go ahead and start. And notice that you probably will say that we're in Grenoble. What time is this there in France right now?
It is 8 p.m.
8 p.m. So it's 12 here in Mountain Time. I can see a little bit of light in that window. Is that true?
It's almost gone now.
It's almost not. Okay. Yeah. Let's
Yeah.
go ahead and start it, please.
Okay. So yeah, the titles, it's kind of like a collection of keywords, right? Synchrotron, nanotechnology, and data science. But that's basically what we're going to be doing, right? So I will first explain what is a synchrotron, why do you use it, what I mean by, I'm not a nanotechnology developer, so I don't work in the development of nanotechnology. So my work is mainly looking at their impacts in environment and in human health, etc. So more assessing the risks. And the data science part, which is the link between Christian or your community and what I do. So hopefully we can see how I've been dealing with the data and what kind of processes we use to interpret and analyze the data. So first, okay, just briefly, just to tell you, so I come from Mexico and I did a bachelor's degree in environmental and environmental chemical engineering, surrounded by nice mountains in the city of Chihuahua. Then later I moved to El Paso, close to the Franklin Mountains, to do a master's in an environmental science and engineering PhD from 2004 until 2010. In 2010, I spent 1 year at the Synchrotron in Berkeley, Advanced Light Source. Again, nice hills, nice mountains there. And then in 2011, I came here to Grenoble as postdoc. Again, here close to the Alps in this beautiful city that I have been enjoying living in here for almost now it's 9 years. And my interests are, so I'm an environmental scientist, so I'm synchrotron, but I'm not a physicist. I'm going to discuss about data science or just present what is my, let's say, my cooking recipes for the data analysis, but I am not a programmer or a data scientist. So these are maybe disclaimers about my presentation. And so my interest is in environmental stress also. You can consider, I initiated my career looking at toxic elements like chromium, arsenic, and in plants mainly, but everything related to environment, so plants, soils, bacteria, microorganisms, etc. And I've been using synchrotron since my first year of master's, thanks to my mentor. And some of the things that we needed at the time, some micrometric resolution, high sensitivity, good beam stability, etc. But then at the same time, towards the end of my PhD, the group I was doing my studies initiated this project regarding looking at the environmental implications of nanotechnologies, in particular nanoparticles. And this is again mainly on plants, but as I came here in France, then I got the opportunity to work not only with plants, but also animal samples, human tissue, cells, etc. So there is really, I have participated in many different projects, all of them very interesting with very interesting scientists, leaders that are doing this research. And my part has been to assist in the collection of the synchrotron data, to learn about data analysis and how we interpret and how we put together a nice story about these, the experiments that they do here. So like I said, my dissertation was mainly on arsenic in desert plant species. And in here, well, okay, basically, I to see this first, where you see colors are attributed to elements. Here we have arsenic, iron, zinc. You can see a little red region here, which is in fact a root which is actually uptaking all the arsenic from the surrounding soil. And with these maps, you know about the distribution. This is the distribution in the region that we call rhizosphere. And this is the distribution of arsenic in what it is really a cross section of a root. And if you, once you have these hotspots there, you can perform these absorption, X-ray absorption curves. And based on this, on the shape of these curves, you can identify the oxidation state of the element of interest. And you can even go and identify the, um, the ligands or the elements which are surrounding that element of interest. So these techniques are called X-ray fluorescence microscopy, and this one is called X-ray absorption spectroscopy. And I'm going to talk more in detail about it. And these are, obviously, these are synchrotron techniques. So what is a synchrotron? A synchrotron light is a kind of electromagnetic radiation, which is emitted by electrons which are accelerated to the speed of light. So in a synchrotron, you first have a linear accelerator of electrons, then you have a booster. In the booster, you accelerate electrons close to the speed of light. You provide them a lot of energy. In the case of ESRF, you provide 6 giga electron volts of energy, and then they circulate in the vacuum in this, what is called storage ring. And tangential to the storage ring, we have laboratories or cabins where we fine-tune this light. We choose certain parts of the spectrum, and then we make them interact with matter, with different samples, and we characterize these samples. What is the advantage of a synchrotron light? And the purpose of this slide is to show you this is what the amount of light in photons per second, per some unit of area and some angle of spreading, but okay, basically photons per second per area that you can get from an X-ray tube. An X-ray tube is something that you will find in an X-ray diffraction, system in a laboratory. There are some in UTEP, there are some in most schools you have these. You have also X-ray fluorescence microscopes, and all of them are based on these X-ray tubes, right? And as you can see, when you go to synchrotron, and there have been several generations of synchrotrons, in the first generation, you have several orders of magnitude of more intensity. So you go from something which is 10 to the 8 photons per second per millimeter square to 10 to the 12. So here you already did a big jump. Then after we created dedicated machines, which are called second generation synchrotrons, where people would go there specifically to perform experiments and analyzing matter. And in here you made another jump. And then around 1994, when the ESRF was built, this is a third generation synchrotron. There is other synchrotrons like this, like Advanced Photon Source in Chicago and the Spring 8 in Japan, which are the first third generation machines. And in here you have another big jump. Then in 2012, at the time I pretty much arrived here in USRF, there was another upgrade, and now we are at 10 to the 20. So you see from 10 to the 8, you can go 10 to the 20. So really many orders of magnitude more light. And now we just completed, the ESRF completed an upgrade, which is called, the project is called Extremely Brilliant Source. And many of the synchrotrons in the world are working on this sort of programs where you are making another jump going towards what would be the diffraction limit, or like the best performance of the machine. So we are close to achieve this like dream performance of a synchrotron machine. But here we're talking now at 10 to 21, 10 to 22 photons per second per millimeter. So really huge amount of light. The advantage is it goes in one very specific direction, and that's how you can concentrate so many photons in such a small area. And with that, you can do a lot of interesting experiments. Now, how is it that you produce this light? Just briefly, there are two kinds of sources. So the reason you generate the light is when you accelerate the electrons at the speed of light, and then you bend them using magnets, then you will generate this, as the time the electron bends and sort of breaks, it will emit a lot of radiation. And this is the synchrotron light. One of the devices that you can use to actually this circular shape of the synchrotron, a bending magnet, and this generates a fan of radiation that you can collect, and you can select the different properties of the light because there is a spread of the electromagnetic spectrum in this cone of emission. If you use another device, which is called an undulator, where you make these pockets of electrons to oscillate many, many times, different periods or different magnets, then you can produce much more light. They produce much more intense light and in a smaller point. So that's just, synchrotron. Just know that what we are generating is light. We use electrons. The electrons produce light. We never make electrons interact with our samples. It's photons, it's light with wavelengths of in the X-ray region mainly, but there's also infrared and UV and ultraviolet that we make interact with our samples. So this is just like synchrotron. Now you know this is photons. It's a light source. It produces a very intense light, and you can use these devices to produce this light. And now the other sort of keyword is what we call a beamline, right? So this is in the side of the machine, and on the other side of this tunnel, you have a beamline. And this is where I do my work. I do my work in a beamline. So when I will say, I was in this beamline or this other beamline, what I mean is an instrumentation that helps us tune the kind of light that we want to use, the kind of radiation, X-ray light that we want to use to produce the characterization of a given material. Now, how many synchrotrons? You may be wondering, how many synchrotrons is there in the world? If you go to this nice website, lightsources.org, you will see a map of the world, and you will see all the points where synchrotrons are present. So you see there's many in Asia, there's one in Australia, one in India, Russia, a lot of them here in Europe. This is the one where I am in, this is Grenoble, there is one in Paris, there is one here in Barcelona, one in UK, and in the US, there is two in California, another one which is in San Diego, which is not very famous. Then the APS and the Brookhaven National Lab and other smaller machines, and one in Canada. You notice also that if we discuss about Latin America or South America and including Mexico, there's only one in Brazil. And they're actually building a new source, but they still have only one operating synchrotron. But there are about 50, yes.
I'm sorry, I didn't mean to interrupt. So I just want to know, how big are these things? Because it's a facility, right? It's a big facility,
Yeah.
where you probably have it there in the slide. If I can wait to see that thing, or maybe I didn't pay attention. But those are facilities, are huge facilities, right?
Yeah.
Thank you. Yeah. So how big are those big facilities?
It depends on the energy that you provide to the electron. This one in the ESRF is a 6 gig electron volt. The energy of the electrons that are circulating is 6 gig electron volts. Okay, that basically has an impact on the diameter of the ring, of the storage ring. This ring here.
So these facilities, they will
840 meters of diameter.
How much? How many?
840. 840 meters diameter. So,
oh, okay.
So this is a large facility. There are some which are small. So for example, most of the experiments I did during my PhD were in the Advanced Light Source, which is in Berkeley, which is a nice little synchrotron, which is in the top of a hill in Berkeley, where you have a beautiful view of the Golden Gate and the Bay Area, and it's amazing. It's up in the hill, and I think that this diameter is probably 100 or 100 some meters. It's a smaller machine, but their electrons only have the electrons have 1.9 giga electron volts. Okay. So it depends. So, but there are large scale facilities. In fact, yeah. Wow. As you said, this one has ESRF has like around 840 meters, and you have around you have like 42 beam lines. Right, 42 end stations, 42 facilities where you can do experiments, and they all work at the same time, 24 hours a day, with the exception of Tuesday when we stop so that they can do maintenance to the accelerator.
Okay, yeah, let's go ahead and see the next slide.
Yeah. Okay, so I want to say what is particular about the European synchrotron, but I work in the European synchrotron, so I have to make some advertisement. But
Mhm.
The thing which is very interesting of the European synchrotron is that this is the only facility in the world that does not belong to a country. It's hosted in France and it operates as a company under the regulations, under the French regulations. But it's actually a consortium where you have 13 member states, where France and Germany are the main contributors, and you have 8 associate countries. And you see all these countries in blue, they contribute to the development of ESRF. And that's pretty interesting. I mean, in general, all synchrotrons are very international, very collaborative environments. They host people from all over the world. You can submit your projects to any synchrotron anywhere in the world. And in principle, they always, if you have a good project, they would accept your proposal. But all of them are run by one government funds. And in this case, is not that. This is really an international facility, and that's pretty cool. I really like this about this way of this kind of governance of ESRF. Okay, let's just quickly say, you know, 17 participating countries. We have about 150 scientists. There is about 6,000 research visitors per year, and the number of projects that they get admitted to try to come and do some experiments there, it's about 2,000 per year. So,
Yeah,
well,
do you mind going back to the other slide? We just want to know what were those gray dots, those locations. I mean, I assume the locations on gray, that means they're synchrotrons, right? But there's some on gray that they're not.
yeah, that's a good question. I don't
That's a
know. I
good
would have to
question.
know. I don't know why there is some of them are orange, some of them are. I guess that perhaps, but this is just. I'm going to answer based on my experience from the. No, it's not going to hold. I was thinking because these 5 here in the US are like the ones which have like public access. And these, it seems to me like these are more, I don't know, I think this one belongs to the National Institute of Standards and Technology, the NIST Institute.
Yeah.
This one I think is Oak Ridge. This one I didn't know it existed. But then again, here in France, this is the ESRF and this is the Soleil. So, and it's a public synchrotron. So I have no answer. I don't know.
That's
We would
fine.
have to go in
Yeah,
the
that's a
website
good
and
question. Yeah.
yeah,
Go ahead.
it's probably, I don't know why they have different colors. Okay, so well, so then why is it interesting to use synchrotron for, in my case, as an environmental scientist? And this can hold any analytical challenge that you may have. If you want to characterize materials, probably it's the same. Basically, like if you start with a pristine material, at this stage, you have a good knowledge of what is your material, and you have generally you have good concentration. So it's not so big analytical challenge unless you're talking about nanomaterials. In that case, you need very good resolution. But then as you try to track this in the release in the environment, and you begin looking at release and fate studies or toxicology or ecotoxicology studies, and then if you then want to try to monitoring them in the environment and in organisms, then the concentrations, they go down, and the complexity of the matrix increases significantly. And so this is really something that puts you on a search for good techniques that can have this enough sensitivity and that can help you to understand these very complicated matrices. So you need things that have good sensitivity. If the materials which are at the nanoscale, you need high resolution. But then there's also another concept which is specificity. I'm going to use this schematic to elaborate a bit on what I mean by specificity. If you really want to know like where a silver nanoparticle is, you can image it, and you can maybe have something which is sensitive to detecting silver atoms. But you're not only interested in is there silver atoms or not, you also want to know what is its chemistry. Usually a silver nanoparticle will begin evolving as it interacts with the media, and it will release some ions, and these ions, they will get in reduction and oxidation processes, and you may end up having, again, metallic silver or silver attached to other elements like chlorine or sulfur, etc. So you would like to have a technique that it's capable to tell you where they are, what are their sizes, and what is their chemistry. This is really very important. And now in here, just like I say, like in the synchrotron, there are many techniques. And here I'm just listing the many beamlines that could help you solving some of these questions at the ESRF. But it's not important, just, you know, so that you know, synchrotron have many beamlines. Beamlines have different techniques, and they all can help you solve one particular question of this problem that you have. In my case, I work at ID21, and the techniques that I use is what is called synchrotron X-ray spectromicroscopy. I use two techniques. One technique is called X-ray fluorescence imaging, and the other one is called X-ray absorption, X-ray near-edge spectroscopy, or X-ray absorption spectroscopy. So if I say SAIMS, I mean this technique, X-ray absorption spectroscopy. If I talk about micro-XRF, I'm talking about X-ray fluorescence. And again, you can see here how it's important that you have specificity, spatial resolution, and sensitivity. The spatial resolution is important because if this is a neuron, you want to know where elements are in the neuron, but you also want to be able to know as more detail you have, like, okay, let's start with the brain section. In the brain section, you maybe want to identify a neuron, and in the neuron, you want to identify a mitochondria. And the more precise you want to be, the smaller the beam needs to be. And that's why it's important to have a beam, a synchrotron beam that you can use, that you can focus to a small size, so that you can produce these images with a high level of detail. In terms of specificity, you can have specificity for elements. So you can say what is iron, what is copper, what is magnesium, but you also want to know whether this is copper plus or copper plus 2, or if it's binding to oxygens or sulfur, etc. And this is something you can get out of SAIMS. And these two, they also have a link with the sensitivity, in the sense that because the synchrotron light is so intense, you can focus beam to a small size, but you can also, since you have so many photons, you can also have high sensitivity and detect in the order of femtograms. So in my experience, of course, there can be techniques that are much better at these, or much better at these, or much better than these, but X-ray fluorescence coupled with the X-ray absorption spectroscopy offers a very good balance of these. And this is the advantage of coming to synchrotron X-ray spectral microscopy. Right?
I think something that caught my attention in previous discussions that I have with Hiram is that if you can go back to the other, with the techniques that you were working, it's like, okay, professor, I remember seeing something like that in my chemistry class a long time ago. But when you deal with this high technology that in the synchrotron is happening, you need to be available to recognize what kind of technology, what kind of components you should be using according to the chemicals components that you're using. And I think that was the next slide that you were gonna discuss that. But I just wanna let because I received a couple of messages right now, they're saying, so what is exactly SAIMS, what is exactly photomograph? So I know you wanna ask those questions.
Yeah.
And those are techniques that, as Dr. Castillo just mentioned, you can just apply it. And you need to have those in order to, depending on the chemist, I don't know if you can see here in the slide, there's our compounds or some chemicals there that you can probably, some chemicals, they get more suitable according to this. So these techniques, and some of them, they're better for other ones. And he's going to explain that one later on, because when we talk about the nanoparticles, some of them behave different according to the chemical compounds and whatnot. So if you're dealing with silver, if you're dealing with coppers, if you're dealing with titanium, so those are some of them there are more feasible to other ones. Am I right, Dr. Castillo, more or less?
Mm-hmm. Yeah, that's correct.
That's it, is what it is. Yeah, thank you.
So that's exactly my point on this slide, is to introduce you to this term, X-ray absorption spectroscopy, or SAIMS, and this idea of using a micro focus beam to produce X-ray fluorescence. Now, I'm going to try to explain a bit more in detail what this means, right? But I wanted to get the message across that a good technique needs to have a good balance between all these, right? You want to be precise where something's located. So if you're talking about nanomaterial, you want to know where it is located. You want to be able to detect this with high sensitivity. So a femtogram is a 10 to the 12 kilogram. So we're talking about like very, very small quantities of material. And the SAINS allows you to say whether this is an element or not only an element, but what is the electronic configuration. So what is the oxidation state and the ligands around. So, okay, let's see. Here, what I want to show you is that when you're using X-ray fluorescence mapping, what is the kind of information you can get? If you go to a beamline like the one I work, depending on the energy of the synchrotron that you are using, between 2 and 9 kiloelectron volts, you can detect all the elements which have color. So I can detect with very good sensitivity from copper to phosphorus. And then if I go in the orange, I have good sensitivity, but I begin to lose a little bit. And that's depending on this transition, which are K, L, M lines. So I won't go too much in detail there. But what I want you to understand is that this is a multi-elemental detection technique, right? You can see several elements at a time. An example is in here. You can see this is a cell from a plant, from the nodule of a plant, where you have a lot of nitrogen fixation. And here's the distribution of calcium, distribution of potassium, and the distribution of iron. And if I am interested not only in knowing where iron is located, but also what is, the chemical speciation, what we call speciation, which is what we probe when we do SAINS of iron, then you can use a monochromator, you can select the line, the light that you want, and obtain these curves. These curves are giving you, the shape of these curves are fingerprinting the chemical status of iron. And the same for any element that you analyze with this technique. So if it's iron metallic, the curve has this shape. If it's iron 3, it has this shape, whether it's oxygen or nitrogen. And based on these shapes, you can distinguish the chemical environment of an element, right? And this can go way to the theoretical development, and you can obtain theoretical spectra of here, but you can do an approach which is very experimental, where you obtain curves from your sample and you compare to what we call model compounds, right? So these are pure compounds that are letting me know where these peaks are located and how they look. And like this, I can do this fingerprinting, right? So that's the combination of X-ray fluorescence and SAINS. And I think I will try to explain a bit more in detail. Again, first, multi-elemental detection by X-ray fluorescence. Here you have, I told you, undulator source, right? So electrons oscillate several times, produce light. We use a pair of crystals to select a specific wavelength, a specific energy of photons. Then they are focused with mirrors or with lenses. And then you have a stage, you scan it, and you have detectors that measure the transmitted light or the fluorescence light. This is what we use for X-ray fluorescence. It's a detector. It's a solid state detector. And basically, when you have an X-ray that comes into an atom, if it has a critical energy, a given energy, which is tabulated, it will eject an electron from some orbital. The electron goes out, and an electron from a higher orbital will come down, and it will emit light. And this is the light that we're detecting here. That's why the way we present in here, they have different colors. The arriving light is one kind of light. The transmitted light is different because it has already interacted with the sample, so it loses some of its colors. And this one is different because this one is fluorescence, right? And this is the fluorescence, the decay. After an excitation, you have decay, and this will emit light. And this light is particular of the elements. So no single element emits at the same time. So how does it work? First, this is what we call an X-ray fluorescence spectrum. So in each one of the pixels here, I have a full X-ray fluorescence spectrum. So if this image has 20 times 100 pixels, and I have one detector which has 2048 channels, this one's got half because I didn't go to the full spectrum, then you will have 20 times 100 times 2048. This is the number of points that you are accumulating. There is a way we would like to reduce this data, right? Because we want to be able to plot it in some way. The way you can reduce it is by creating a region of interest. And so you select this peak, which we know corresponds to an element, We select this peak that we know corresponds to another element, and then this peak, and it corresponds to another element. What are these elements? Here you have titanium. You have to trust me. With time, you learn where they fit. This part is not calibrated, but we can calibrate. We can convert channels to electron volts, and then this will guide you more as you become more experienced with fluorescence, X-ray fluorescence spectroscopy. So then basically now what I have is 20 times 100 and some value here, the integration of this peak, and some value here, the integration of this peak, and integration of this peak. And then I can superpose them, and I have this distribution of titanium, calcium, and potassium. And you can see now the shape of a nice cylinder of a root. So this is a root. Later I will show you more details about this application. And if I want to see only titanium, then I can maybe, instead of putting everything in red, I can just assume that where the highest intensity of titanium is found, I will see red, and when there is almost no titanium, I will see blue. And now this is a different way of representing the data. I am only looking at the integration of this peak, right? So I have intensity in each one of these pixels. And now, okay, now I know that this titanium is located in these walls of the root of my interesting sample. But I wonder, what is the chemical information? What kind of titanium chemistry I have in those spots, right? So I have here marked some regions of interest where I can go and put my beam there, for example, and then I perform this scan, and I will get a curve with a shape. Here I have curves with shapes of the main chemical species of titanium oxide, anatase, and rutile. This is the way they are configured, the way they pack. And you see that you have different features. You have some differences here, you have a difference here, you have, right? You have some features that allow you to distinguish when the chemistry is rutile and when the chemistry is anatase. And based on that, then you can say more about, you don't only say there's titanium there, you can say there is titanium, but in this spot I have rutile, and in this spot I have anatase. So now you have learned more about your sample.
So something that caught my attention on this one, and I'm not so sure we're gonna have time to go through orange, but eventually there is a software to basically tells you more or less these distributions. I like this these slides in particular because for all these experiments that Hiram just mentioned, you have the experts like Dr. Castillo, who said, like, you know what, here is something that we can have high observation or high spectroscopy for titanium or some whatnot. Here's where the experts can make the multi-criteria decision making based on the results of the fluorescence results that we obtain. And there's some software for that. I don't want to anticipate what's going to come later on, but I want you to pay attention on this particular slide, because here, this is what you usually do in a regular, well, not in a regular lab, but here you can have this power to extract that resolution of the information. Later on, we'll see there's some specific software where you can actually extract that information and make the selection based on what you need or based what the expert says it has.
Okay. Yes. So okay, we can continue. So okay, now I hope that that the idea is more or less clear. When I mean X-ray fluorescence and when I mean X-ray absorption spectroscopy, X-ray fluorescence are basically these images, and you will see here elements that correspond to given colors, and these are the curves that allow me to say a bit more about the chemistry of the element that I am interested in. So I'm going to start with, before I go in my big speech about nanoparticles in plants and all that, and a bit of how the amount of data that we're collecting is increasing with time, I want to catch your attention with this one, which is, this is a real material, right? ActiCoat does exist. You use it in the clinics. It's in order to treat patients that have severe burns. So they have very big wounds, like a good proportion of their bodies has been wounded by burnings. And if you put the dressing, because silver has the capability to prevent bacterial growth, so it inhibits bacterial growth, so it's a good antibacterial and antimycotic agent. And it does that by releasing ions and creating eliciting toxicity over these bacteria. It can have some toxicity also in humans, but it's not the same level as the bacteria. So if you put this coating, you prevent the infection, and that helps the patient to heal faster, just by preventing the infection. So what we did is we wanted to understand, it's a collaboration with Marco Romano, an Italian group from the University of Venice. So we wanted to see in patient biopsies. So here you see the biopsy, and here you see the square where we did the map, and here you see the distribution of silver, and then we use phosphorus, sulfur, and chlorine just to show you how is the structure of this skin biopsy that we have. And you can see that in the epidermis, which is now severely wounded by the burn, you have a big deposition of silver. So there's a layer of silver, and as you go more in the internal the dermis and the different layers of the skin, then the silver signal disappears. So we wanted to understand the chemistry of that. What do we have in here? Is these are still silver nanoparticles, or is this something else? So we took several spots from the red towards the less concentrated regions. And what you see again, if we use this fingerprinting approach, I want you to pay attention to this silver chloride and look at this peak in particular. So this is the way the spectrum of silver will the shape of it when it is found as silver chloride. And you can see here you have in most of the spots where you have all these very high intensity, all these red regions, we have mainly silver chloride. As you move away from that part into the more diffuse areas, then you have this feature disappears, and you can compare it to this one. And this, what I mean here with AGGSH, is basically silver glutathione. Glutathione is a molecule that exists in the cells, human body, and it helps you to detoxify heavy metals. It links to things like cadmium and mercury, and it helps you to detoxify. So it means that you have a crust of chlorides. Some of the ions are leaching, but they are trapped by this glutathione. It seems like it's the big picture here. And the good thing is that this we did after 3 days or 4 days of treatment with this coat, active coat. And then my colleagues also got biopsies of 12 days after. You see there is no longer silver. The epidermis has recovered. You see the nice combination of silver, chlorine, phosphorus. It tells you how things are. You have a vesicle there. And there's a little bit of reds here and there. It's only detected by X-ray fluorescence. We couldn't do any of this spectroscopy analysis, which requires higher concentrations of the element. But basically, in summary, we see it really helps to prevent infection. Then the wound heals, and the silver signal disappears. So part of it is probably washed off the surface, and whatever comes in is systemically eliminated. Can you do this material maybe a little bit better? Yes, perhaps you can maybe fine-tune its design, its engineering, so that it does not release so much silver. I don't think it is required, but okay, in overall it works well. And you can see how these kind of materials, we are probing them as they interact with human samples, right? We are capable to see how they work as they are operating, right? As they are doing their work. So that, I thought that was an interesting application to show you. Another one regarding nanomaterials, which is this one is what we can call its own purpose, right? We create the material and we deliberately expose someone to this material. We know that it has to be a nanomaterial, and then we get exposed because there is a benefit after. But in here, what we have is nanoparticles in tattoo pigments. In principle, people does not know, are not sure that there are nanoparticles in the pigments that are used for tattoos, right? In this case, the titanium oxide is used in white powder, and you can mix it with different colors to create different tonalities of colors. For example, if you have red and you put white, you will get a pink color, etc. So it's quite often that you use this white powder, which is titanium oxide, and you mix it and then you use it in tattoos. And the question was like, okay, so we know that the tattoos are pigments and they will be in the skin. So you can get biopsies from, these were patients, these are corpses. So you see already the pigments, right, with normal microscopy. You can use also some labeling, which is DAPI, which is just telling you where you have a nucleus or of cells. And these are X-ray images. Again, this is X-ray fluorescence. You have bromine, titanium, and phosphorus. You see big chunks of red, so a lot of titanium where the pigments are present. And the question was like, okay, but I inject this in the skin, so okay, it's not a problem, right? But the thing is like your immune system is very, very smart, and it sees a foreign object and it tries to eliminate. So it gets collected and it goes more or less to the lymph nodes, right? And you accumulate this in the lymph nodes. You see also in red, the titanium, where you can already see the pigment, which is kind of greenish in this case. And you see it collocates with bromine because in the molecule that you're mixing, this green pigment, which is mixed with the white powder, contains bromine. So we are sure these are the pigments. So your lymph nodes, okay, the lymph nodes of this person are contaminated with the pigments that he put in his tattoo in the skin. Another thing that we got from synchrotron nano imaging is the size of these particles. You see here, this is 2 micron. The pixel size is 50 micron. So these particles, they are in the order of 150 to 200 nanometers. And the smaller they are, the more you can bring from the skin to the lymph node. So the mobility of these pigments depends on their size. And that's pretty interesting. And another question that my colleagues had is, what is their chemistry? Is this anatase or rutile? Because we know that in these pigments, you will use either anatase or rutile. In general, what you use is rutile because for some reason it's considered to be more compatible with, biocompatible, let's say, compatible with your body. And if you look at the curves, it's not labeled here, you'll have to trust me, but this split here, it's a characteristic of rutile. So we have 4 spectra that we found where there was rutile and some of them anatase. In general, most of the spots where we tested for with X-ray absorption spectroscopy, we found rutile, but there are some of them where we found anatase. And anatase is photocatalytical, it's photoactive. And so it's not a good idea, let's say, to put anatase particles into your skin. So no wonder why the majority is rutile, but there are some of that's anatase, and this might be related with allergies to tattoos, etc. I don't want to go too much into like overstating or overinterpreting the data, but this is basically what we found. Like they move from skin to lymph node, they are small and they are oxides, right? So they are titanium oxide nanoparticles. Okay, so well, then this was more like related to human health, but we know applications of nanotechnology, and I put this one, there are so many in electronics, industrial. I'm not going to try to cover too much, like which nanoparticle is used in what, but we know there are so many applications like nanotechnologies in the last, I don't know, 15 years or 20 years, they are in boom. They are produced in significant quantities, and more we try to make more smart devices, etc. So there are more applications. But one of the questions is like, if you produce a material, you want to make it compatible with the environment. And that's where my part comes, right? Because I'm an environmental scientist. So this is the part I am interested. I'm not so much interested in developing new materials. And you will see there is a way you can combine the 2 aspects, right? Like the environmentally friendly or the reducing the toxicity, and developing a material at the same time. But in my case, it's like materials that are already produced. I want to know what impacts they have in the environment. And in particular, because I show you, I did my thesis on plants, and Dr. Gardea, who was my supervisor of my dissertation, is always working in plants. So if you look at this review, there are nickel particles, iron particles, cerium, titanium. We have a lot of questions about how they interact with roots, how they go into leaf, into the edible parts of the plants, etc. So I started working on these a little bit towards the end of my PhD. So my advisor, Dr. Jorge Gardea from UTEP, he got involved in this very big project with the University of California. And part of the work of the group was to determine what are the effects of these nanoparticles to plants. So the first experiment that I participated, so I was still a PhD student, but I collaborated in this, so my name is hence in the paper, was about what kind of modifications cerium oxide and zinc oxide nanoparticles they have in soybean. And we did a very simple experiment. In here, you see there are no images. It's just X-ray absorption curves. And what we found is that zinc oxide nanoparticles, but not cerium oxide nanoparticles, are dissolved or transformed in roots of soybean plants. How do we know that? Well, here you have cerium nitrate, which is basically cerium plus 3. And this one with the two, like looks like a camel, right? With the two balls, the two heels here, it's cerium 4 or cerium oxide. And basically, when you compare the dotted line, which is coming from the soybean roots, and the full line, which comes from the cerium oxide nanoparticle, you don't see a significant difference. So this leads to a conclusion that they are actually not changing. They are capable to get into the roots, but they are not changing. And this is how you do that. You get many roots, you dry them, you smash them, and you produce a powder, and you take an overall sample. There is no spatial information in this experiment. If we do the same comparing curves of zinc oxide, you see how you have many like very sharp features. Whereas when you look at the one that comes from the root, it has like one big white line here, big sharp peak, and then some oscillations. So you can compare these not to the zinc oxide nanoparticle, the pristine material, but mainly to something which is acetate or nitrate, which means is mainly ionic zinc, right? So that's what it's leading to the conclusion that zinc oxide, but not cerium oxide, is dissolved in the root of soybean. And this is only X-ray absorption spectroscopy. And I want to say in terms of data science, basically we have 4 or 5 spectrum here, 4 or 5 spectrum here, and we can, I rapidly did the analysis in front of you, and I think that you more or less, I hope that you trust in the interpretation that we did. So this was simple and not too much data, right? And this was back in 2009. But then in 2010, I was doing my internship at the advanced light source, and now we had the possibility to do images. So we said, okay, let's do images. So I contacted my friend, this is his dissertation, so Jose Hernandez, and my other friend, which is Christian's sister, Alia Sabin. And we produced these samples, brought them in the synchrotron, and this is a leaf. You can see the veins of the leaf. This is the plant, mesquite. And so we mapped a part of the leaf, and in this case, I'm displaying zinc as green, and you see spots. And now what I need to know in order to investigate the chemistry is to take spectra from several spots, and we classify them more or less into diffuse spots, which belong to like a kind of diffuse scatter signal, and then hot spots, which are coming from the very bright spots. And we collected, I don't know, maybe like 20 spectra. And then we came up with this figure showing basically the same thing as we did in soybean, that the zinc oxide nanoparticles, they enter into the mosquito plants, the zinc goes up into the leaf. There is more zinc in the treated plants than in the control, but we don't see signs of any nanoparticles. It seems like the zinc oxide nanoparticles, they get dissolved by the plant. This is now a combination of X-ray fluorescence, where you can see where elements are, where zinc are, and then many, many curves of zinc, X-ray absorption spectroscopy. And this is what we managed to do in 2010. At that moment, I want to say that in terms of the complexity of the data from imaging, and I'm going to come back here, there is 2 ways you can do this experiment. You can collect the full spectrum X-ray fluorescence from each one of the pixels, or you can just, before you do the experiment, you say, I know this is zinc, I know this is calcium, I know this is potassium, and you create this region of interest, and you are already minimizing from 2048 channels, you minimize to, I don't know, 9 elements or 8 elements. The problem is, if I created a region of interest here and here and here, but I didn't create in here, these peaks, I don't retrieve them anymore. They are lost because the data was not saved. And that's the way we were capable of doing experiments at that moment. We could only have elemental distributions. We didn't have full spectrum where we can do this sort of fitting that I will show you how it's done after. So that's in terms of like, you know, the data, right? The kind of data that we're getting, how we process it. This was already kind of reduced data. We have elements, maps of elements. Okay. So well, then then I graduated and I came back as postdoc here to ESRF, and I continued collaborating with Jose and with Ali as well. So this picture of Jose. And we did, again, we continued on soybean, but we wanted to know if these particles were capable to go all the way up to the soybean grain. So overall, I mean, in summary in here, we managed to detect some cerium oxide spots in the grains. So this leads to the conclusion to say that when you put nanoparticles in the soil, in cerium oxide, and you grow them, you will have some of them reaching the soybean, which is the edible part, and that's up to this point we stop. We cannot say is this toxic to the plant or to the human health or whatever, but we know there is transfer, right? So we're looking in the environment. We want to know how things move around and at what moment we can find potential toxicity or some environmental issue about it. So this was very interesting, and it was a combination of ESRF where we did all the cerium work, but then there was more. These are nice images of zinc distribution in roots and in the beans, and this was done at the Stanford Synchrotron. So that's why you have the logos of the two, the California Stanford Synchrotron and the Grenoble European Synchrotron. At this moment, now we have in ESRF, in each pixel, a full spectrum. So now, after I collect the data, I can go back and get the full spectrum from each one of the pixels here and make a fit or make a ROI attribution and recover the image. I mean, I have the full raw data, and I can reproduce the images if I am not happy with the way I fitted them, if I am not happy with the way they, for example, you can observe here, there is a crosstalk between this one and this one. So if I want to deconvolute these signals, if I want to fit them and attribute really how many counts go to the calcium, how many counts go to the potassium, I can do it by taking this approach. And this approach can only be done if you're saving a full spectrum at each one of the pixels. And this is what, in my career, I don't mean it was the state of the art. I know the beamless are probably doing the same thing, but at the moment I arrived in ESRF, for me, this was a big leap. It was a big change now to have like all this data there that I can, if I forgot about one element and I find that I can go back and retrieve this image. And that was pretty cool to have. Okay, so now I'm going to discuss a bit more about titanium dioxide. So I just wanted to remind you that as we see that they're using in paints and coatings, as it was the case on the tattoos, they are incorporated in plastics, in cosmetics, toothpaste, in food industry, so a lot in bubble gum, in a lot of candy for kids, in the bakery industry, photovoltaics, etc. Titanium dioxide is used in so many applications, and what we are trying to understand is, okay, you produce them, you use them, and then, for example, if it's a sunscreen, you wash your body, and then it goes in the water stream, and then it goes to a wastewater treatment plant, and then at some point it comes into a landfill, or perhaps you use the waste, I mean the sludge, and you put it into an agricultural system. So how they move in air, in water, in soil, what happens after their main use, right? Either in a sunscreen or in a painting or in a cosmetic or whatever. So this was also back in 2012 when it was one of the two very nice experiments that I did. This was the PhD project of Alia Servin, so it's Christian's sister. And in here, what we were able to say is that if you put cucumber plants in presence of titanium oxide, you will see that they can accumulate in the root. So again, these are spots, these are the curves. And these features here are trichomes. These are hairs in the leaf of the plant. It's like they have these big hairs in their leaf. And this region corresponds to this region. So in the basis of the trichomes in these hairs, you accumulate a lot of titanium oxide nanoparticles. And what was more interesting is that this was exposed to a mixture of anatase and rutile. And again, you have to look at this split right here. You have to look at this feature here, which is rutile, which is absent in anatase. And it was very interesting to see that what you here, you find a majority of rutile, and in the root, you find a majority of anatase. And to this point, I don't have really an explanation why is that, but the plant is capable of doing this, and I thought that was a pretty, very interesting result. We also analyze the fruit, so very small cucumbers that Dalia sent here in the lab, and we make analysis, and we found also traces of titanium oxide. So there is evidence, not only from these experiments, but from other experiments in a lot of papers in the literature, that plants are capable of uptaking titanium oxide particles and bring them all the way to the leaf and the edible parts. Now we have also evidence that alfalfa is capable of doing that, and alfalfa, you use it to feed cows, etc. So there is a kind of like a circle in the environment of these titanium oxide particles. And that's what led to this study. Now this is the work of a postdoc, and Elena Pradas from Madrid. She was working in a wastewater plant, a pilot plant, where they were introducing silver nanoparticles, in fact. But when we analyzed the silver, we found that there was a lot of titanium oxide. This is a real stream. And the sludge, it comes from a pilot plant, but the water that goes in is a real stream from a city in Switzerland. And we found a lot of, you see now here, the red is titanium. So we found a lot of titanium all around the silicates and surrounding organic matter, etc. So the sludge contains a lot of these titanium oxide particles. You can see images from transmission electron microscopy. And now this starts to get a bit complicated, because now we produce many Saints curves. And this is a way of presenting them. Now, if you compare, when I present you this, then you can see, yeah, the features now. If I present 5 curves, you begin to lose a bit the trend, right? Because now it's not so clear. If I present you all these, now it seems like I'm kind of maybe hiding some of the information, right? So you have to generate a table, and you have to find a way to present this data, because now we have probably collected like about 60 to 100 spectra from different, from only one sample actually. And so in general, what we found is that based on this speciation, it's split between anatase and rutile. That's an obvious result, because it's more or less used in both forms in real nanomaterials. And if you look in the sludge, you have evidence that this is really coming from nanomaterials, because they have these very sharp symmetries, right? Very sharp faces. Whereas if you look in soil, you will find oxides as well, but they have this grainy, very amorphous kind of shape. And we're still trying to find, like, how can we distinguish the nanomaterials we produce from the titanium oxide, which is naturally abundant in soils and in water bodies, water systems. So that was one question, and this led to another project, right? So I said this is sludge, and this sludge in all over the world, you use the sludge from wastewater treatment plants, and you amend fields with it, because it has a lot of organic matter, it has a lot of important nutrients, but it also contains some toxic elements. And titanium oxide is not regulated, it's not even studied in depth, like how much of this is in the, or how much is nanos, etc. So this is starting, is like a upcoming element of interest, right? To investigate whether it's toxicity or not. What happens when you put in the fields, if it goes in the crops, like I show you from other experiments that we have, some evidence that it moves in the plants. But we wanted to see like real life cases. So this was the postdoc of my friend Juan Reyes from Mexico. And we went to Chihuahua, so El Paso is here, and we sampled 1, 2, 3, 4. This is the 4 wastewater treatment plants that produce sludge in Chihuahua. So basically we sampled all of them. And then we applied a kind of complex like multi-technique approach where we use X-ray fluorescence and synchrotron techniques, nano and micro scale, and also some other classical techniques from laboratory. But when we began studying, so we produced this pellet of the sludge, and you see like all these tiny spots, and then some wide regions which make a few or several microns. And you see all these spots there is where we are collecting SANEs. So now we are really beginning to collect more than 100 spectra. Actually, we have close to 1,000 or more than 1,000 spectra in this project because we did several pellets from the different wastewater treatment plants. And we decided to do a classification. We classify the spectra, I mean, we put a tag on it, where when we have large particles, so in here, so I put a tag that says large. If they come from spots which make less than 3 micron or smaller, we have a resolution of about 500 nanometers in the beamline I work. So we call them small. So there's large and small, and we wanted to see if there was a trend. In here, I'm showing you already the result. The trend is that in the large particles, we have this kind of spectrum, which belongs to a naturally abundant ilmenite crystalline phase or an amorphous kind of titanium oxide. And in the small particles, these, and if you remember, there's no split here, so this is anatase. So we were thinking, okay, it's in Chihuahua, well, sunscreens and coatings, that's likely the origin of all these particles in the sludge. So here, this is what Christian was saying. So I'm telling you now we are in 2019, and now we are in this project where we have had the opportunity to have a lot of beam time and to do very, very fast SANEs, a lot of maps. And now we have a data set, which is like 1,000 spectrum. And we have to exploit it somehow. And so this is where we begin using, this is a print screen from software called Orange, which is an open source software. And you see the kind of programming that you do is like some like good data scientists, they produce widgets. And then we, like the very knowledgeable ones, we collect these widgets and we link them in some kind of a workflow, and then we get some result, right? So in here, we first have some pre-processing where we use either second derivative or a normalization using vector. And then we try to produce, in this case, we use logistic regression. And so basically, out of these confusion matrices, we see which ones are classified in the spectra as small, as large. And at the end, our hypothesis was correct. So some spectra which was misclassified, we managed to extract their spectrum, and it's like this. And here, you see this split here, this is rutile. So the ones that didn't fit into this small, large, that they were a bit outliers, we found that they were rutile. And in overall, for the ones that are all coming from the small points, we have like dominantly the presence of anatase. And in the ones which are large, we have mainly this spectrum, which is this ilmenite kind of amorphous titanium oxide. So in here at the end, like I'm basically reducing my data to 3 spectra, and I have more than 1,000 spectra. And that was pretty cool. This workflow was developed by Juan. So fortunately, so I don't have this one. I'm going to show you a little bit how I do with another data set, not precisely this one. And in summary, we managed to prove using nano X-ray fluorescence that we have titanium oxide nanoparticles in the sludge from the 4 wastewater treatment plants in Chihuahua. We know that dominantly all these small spots are anatase, and that the big chunks here that we see, they correspond to non-well crystallized titanium ions, or something that comes more from the environment, right? Which is just maybe washed off by the water effluents, etc. So we have identified very clearly these 3 different chemical species of titanium. And that's the summary of it. Using Orange was key for making sense of this data, because if I try to plot in this way 1,000 spectrum, well, it's not feasible, right? it doesn't work. So you need some way to find these features and to make this identification, classification. So that's where I think this is the data science part of my of my
Yeah.
presentation. Um, Finally, I want to show you this. This is a mode in which we are moving towards. Is in fact now, you see the image is 50 times 70 pixels. I use one detector with 248 channels, but the problem or the interesting thing, the reason why you see a movie is because I repeat this image 100 or 200 times. So I have a stack of images that need to be aligned so that I can always have this spot in there. And basically, if I repeat the movie, you see in green you have silver. As I am changing energy and repeating the image, then you see the titanium spots, they appear. And what you see here is the sum or the mean of all the red pixels. But in reality, what we want to do is we want to be able to go and get individual spectra from each one of the pixels. And when you manage to do that and you manage to expand the field of view, then you can have an idea of the size of the number of spectra that we're going to get, right? The size of the matrix that we're going to have to process. So again, you will need these machine learning algorithms to help you interpret all this data. In this mode, we're working towards like, how are we going to do that? Well, there are several ways. First of all, you can make this pixel smaller. You can reduce the amount of time that you count. You can multiply times 10 the number of detectors. And this is what we are trying to do, right? If you multiply times 10, then you can decrease the amount of time that it takes you to do this image. And if you decrease this time, then you can invest time in expanding the area that you are analyzing, right? And that's kind of like it's kind of the state of the art is where a lot of beamlines are working now. Some of them are capable of doing already kind of large field of views, and also we're more or less in this size.
I think something that I think is worth mentioning, and I think this is a great presentation. Let me just tell you something that we need to unpack here, a couple of items, because the whole idea when it's very interesting, and I'm glad that you put a couple of those pictures that was familiar with me, familiar in the sense that very familiar with me. The work that you did with Alia, I remember when she was working about titanium, and is widely used in many of the products that we use every days, right? Cosmetics, you mentioned things like that. But the question is, what happens after you use them? And I think that slide that you have, when you just throw it, or you can just wash it, and it goes to the environment. There's a big impact in many different aspects. Environmentally speaking, people actually for many years kind of forgot about the environment. Right now, we're not in the position right now to just omit what is happening to the environment. Obviously, we have a crisis in the environment and we need to pay attention. Before going there, because there's a couple of things that I want to talk about this in your presentation, is that before going to nanoparticles or the synchrotron or whatnot, everything is composed by some material, by some components, by some chemistry, but we never pay attention what is the impact that they have. I mean, I receive a couple of messages right now, private messages, like what are the negative effects of using synchrotron, or what if we use silver, what is the impact of silver in the blood and things like that. So there was a couple of messages that I'm reading there, but which makes a lot of sense what you're doing. Sometimes when we process items and we don't have the ability to identify what kind of components are there. So what we can do right now with this technology, first of all, we have nanoparticles in one side, which allow us to go and move and identify the components. Because now with nanoparticles, you can actually extract 10 to the power, what was this, 9, or I think it's -9, something like that. You can visualize things in more detail, what is happening. Second, we can see the transition between one element, with one material to another material. It depends how it gets transformed. Third thing, since we're dealing with data which is very minuscule, we need the power of technology like the synchrotron. That way can visualize things in a more higher spectrum. Of course, the mechanisms that we're going to use, they're not just put it there in the synchrotron and let's see what happens. You need experts who will be recognizing the part, every single stage, what happens from stage A to stage B, from stage B to stage C. Are they transitive? If they're transitive, what is the degradation per state? So all that data is impossible to measure in just in a regular lab. So all that data will be get stored in somewhere. And I think that's where, if you don't mind just going to the slide of orange real quick, because I think this slide, when you talk about in the aspect of orange, orange is only a software, yeah, that you basically drag and drop a lot of items like this. Thank you. This slide that you see here is just composed by different widgets, what they call it. Okay, so in reality, you can just bring databases, you can bring a lot of information there, but I don't know if you can see, notice that there's some connectors, there's some edges that connects these vertices, and every vertice is a mechanism, right? In other words, what you see right now is an algorithm. You see the saints, yeah, from the pellets that you receive, probably some data, right? And then you see that arrow that goes through this saints and then goes to the data to raw spectra. For that, you can extract some information and transform in some data. And there's some branching, right? Where you can just go to a spectra 2, or you can just send in that information, some data sample. And from there, you can start conversion. All these mechanisms, they're called algorithms. Yes, professor, but the algorithms, I thought you just put it in the code. Yes, of course. An algorithm is a sequence of finite steps in order to solve something. And in some of them, you can see those kind of distributions there using some graphs, and that's where the analysis goes. And if you go to the next slide, I like this slide that you just put here. Thank you. When you see the routine, you see the interpretation that goes there, but there's some frame around that is the boundary. It can be an error, or it can be something that actually oscillates up and down. You have upper bounds and lower bounds depending of the spectrum that you just got. Am I right, Hiram? Is that what you're actually saying? So the
Yeah,
experts is basically when they talk to the client, and let's just talk about clients, just not someone who's going to purchase your product, but someone who has an interest to identify, is it okay if I use this more? Well, the upper bound, you cannot get this part. The lower bound, you're going to get that part. And the expert is what they go to industry right now, or national labs or whatnot, to identify what will be the best case and the worst case scenario. We're just talking about one spectra or one item, but when you talk about trillions of databases, right, it's impossible to measure. And Iram just mentioned, at this point we need help, maybe from more systematic way to do it, or maybe through artificial intelligence elements, or some kind of training that he said, you know what, this is getting out of control, or you know what, this piece of information has a lot of X, Y, and Z. So here's when we have the intersection between the environmental engineering, when we have a lot of different items, and we can go back to the discussion that we have previously. When you want to get something involved with nanoparticles, for example, if you want to do a device that deals with nanoparticles, what happens when you throw it to the trash can, right? It's like it doesn't work. How do you recycle it? What is the impact? So many people might say, oh, with this, we're gonna get this amount of money, but you know what? The impacts on the environment is gonna hurt you badly. And here's when we need to pay attention in the next generations of scientists. When we design a new software, when it's not software, hardware, or maybe when we want to go and get some crops for certain lands or soils, what exactly we're putting on Earth, and what is the implication maybe in a couple of years, maybe in some decades? What is the impact of later on? You also talk about items that you actually introduce to your body. It will help you, right, at some extent, but how long it will be helpful, until what time, and if we're going to have some effects. One of the questions is about what are the negative effects of nanoparticles, and I'm going to let you actually talk about, I know more or less where you're going to go talk, but I want you to discuss that a little bit more. If you can just discuss that having the nanoparticles later on. But that's basically what I wanted to unpack here at this point, because everything that we have been discussing, it makes a lot of sense. If you're in the environmental engineering, when you say like, oh, this makes sense, let's do this, let's do that. But when you have a lot of data, a lot of components, everything put in together, is impossible to make it just one decision right away. We need help. And the help comes from maybe people who are using this technology, like the synchrotron people, the scientists and the engineers who are working in this facility can help us. Of course, we cannot just go there, hey, can you please test this? There's thousands. He mentioned at the very beginning, there's more than 2,000 proposals that they get submitted per year, things like that. But I think that's something that we should consider, that the ethical implications when we have some kind of a product that might impact in nanotechnology. Hiram?
Yeah, yeah, sure. Yeah, that's I think always when you produce a technology um, or a material, you have to think about the performance in the application that you are um, producing this material for, with what purpose at the moment of use, but also what happens after. So it's what it's called cradle to cradle, right? Before it was like
Right.
cradle to grave. And basically, we would stop reflecting on things and impacts as we dispose of the material. So we create, we use, we dispose. But now we don't have enough space. We need to reuse, we need to recycle, we need to make sure that we complete the full cycle of a material. And in that sense, for example, the fact that these titanium oxide nanoparticles, they end up in the sludge, so far I don't have, from our studies and from studies that I know, maybe I'm wrong, I don't know, maybe someone will call and say, yeah, no, but you know, there's this, that, but so far we don't have strong evidence that says that this is really posing a problem to the plants, but or to the soils. But I don't think the study is fully done, right? And if that is the case, and there is no bad impact, then we found a way to complete the cycle, right? Because these titanium oxide particles, they wash off of our paintings, coatings, of our sunscreens, and then they go in the wastewater treatment plant, and then they're in the sludge, and we put them in the agricultural field, and that's like reusing, right? As long as there's no negative impact, then we don't have a problem to continue doing that, right? So that's what we need. That's why I really like this project, because I think we need to investigate in depth what is happening, because we are probably at the moment where we can still maybe stop or revert any environmental issues that we may be causing, right?
And
So
something
and
that, yeah, I was
another
gonna say it real quick. Now that you mentioned about this kind of problem, the impact, and I noticed that you said that this synchrotron is mostly funded by most of the European nations, like Germany or most of them, right? That, do you think there is an impact, or these countries are more conscious about other impacts in the environment other than others, or why do you think has more funding for those particular countries, particularly because you deal with nanoparticles and items that affect the environment? Do you, or maybe we should not even mention the politics involved
yeah,
behind this sense, but
yeah.
I think they have some kind of consciousness here in creating this kind of facilities that we can actually investigate. You mentioned to go investigate what will be the impact in the next months or next years.
Yeah, and actually, I mean, I want to use this last slide of my presentation, because I won't have any more than the acknowledgements, and then if you want, we can see
Sure,
everything.
sure, sure. Go ahead.
But because in that regards, and also about a question that I think you received before even the podcast, like I like to use this application. This is a collaborative work of people working in synchrotron, people working in material science, people working in biology. So the persons that did all the cell culturing, exposing, characterization of the cells, the guys that produce from here in Grenoble, from CA, that produce the nanowires. It's really a huge collaborative work. I have a small contribution because I was beamline and I helped them doing the, in particular, the part you probably can know if you see these plots, right? And these images are coming from ID21. And in here you have all the, why is not working? There. You have another a coupling of X-ray fluorescence with nanotomography. And this is a fibroblast cell. So what is the purpose of this concept, safer by design, is what I would like, in this way, I would like to see most of our materials being produced in a way that you produce a material that has good functioning, the good properties that you need for your application, but it has minimum risk after its use, or if it's or if there is failure, right? So in this case, the idea was, okay, we have a functional material, we have nanowires that we want to use in flexible cell phone screens. So we need a certain conductivity. We need that when we touch, the signal is communicated, etc. So you can have that functioning material at 90 nanometers diameter or at 30 for the nanowires. You can create a network that has properties that are good for producing these screens. And the question is, if you play on these parameters, what happens to the other part? What happens when the screen in any way is flexible, but it might break, or someone might like on purpose break it with, I don't know, with a hammer or something, or it falls, or you say, I no longer want this cell phone and I'm just going to put it there and they will try to recycle it or just put it in the landfill. What happens when these nanowires get released from these devices? If you study the 30 nanometer or the 90 nanometer, you will see that if it's 9 nanometer wire, it will go in the cell. So this is like a cell here. It will go in, and in fact, it's going to be capable of operating as a kind of a lancet, right? It will go through the cell, it will go through the organelles inside the cell, and the cell will have an oxidative burst. A lot of reactive oxygen species, a lot of silver ions being released, enzymatic processes, and the cell which is going to basically burst up, dies. If you have 30 nanometer, it goes in and it starts rolling. And it rolls and it gets inside this vesicle, which is the vesicle which was the mechanism how it went in the cell. And then the toxicity is lower because now the cell knows that there's a foreign object in a given pocket, and it will try to do chemistry. So attacking these ions, releasing, producing some sulfidation. This is what we saw from the SAMs. It starts getting sulfidized, some ions are released. You have glutathione in there as well, binding silver. And the toxicity is much lower if you have 30 nanometers than if you have 90 nanometers. And this material functions as well as this one. And that's pretty neat. That you look at parameters that you can optimize. You don't sacrifice functioning, or maybe at some point you would have to sacrifice functioning in order to make your material safer, right? But it's a balance of the two things. It's a balance of the purpose of the material and what is going to happen once you don't use it, or if accidentally you break it, or it's just not working anymore, right? Or someone touches a screen which is damaged, right? And you get your skin cells exposed to nanowires, etc. What happens? This is a bit the question. And I really like this application because I mean, there are very cool synchrotron techniques and I like the images, but that's probably because I work in synchrotron. But I think that the full story behind it is very interesting, right? That you can actually fine tune your materials and reduce their toxicity from the beginning, and not once you put them in the screens and then you see, look, it's toxic. Now what do we do? Why don't we go back and try to make it again? But at the time you already produced, I don't know, 1 million cell phones, or it's kind of late. This same thing happened with asbestos, for example. We waited until like several years after we saw people dying from asbestosis. And then people was like, this material at the end is not as cool as we thought. is really, really toxic. Now we ban it. Now we recover it. Now we, I mean, that's late, right? You want to do things ahead.
Yeah, this is definitely a very interesting application. I mean, it's at some point we need to balance in what it will be the toxic part or not the toxic part. I have a question here that it says, you mentioned computational quantum computing applications. Can you mention one of those applications with nanoparticles? Do you know anything by any chance?
No, not really, not
No,
much.
it was there also. I noticed that there was quantum computing there, but
Yeah, I mean,
I'm not
yeah,
so sure what exactly. I mean, right now there is very popular. I have another question. Oh no, that was another. It was just a comment from. Yeah, thank you, Alia. Yeah, yeah.
but
This
this is
is
some.
okay. What is this, Ida?
Okay, yeah. I mean, I don't know. I don't know if there's other questions. If you want to discuss other thing, otherwise, I don't know.
No,
Maybe
that
show
was a
you a
comment
video.
from my sister, who was hearing
Hello,
that.
hello.
What Hiram is showing us right now at this point, it's Orange, is the software that he was talking about in one of his slides, and I'm glad that you were able to pull that up real quick because we have a couple of statisticians here in the forum right now, which makes us feel a little bit scared. But can you please explain us this?
Yeah, okay. So this is a data set that I've been working on, and we're getting it ready for papers. It's not published yet. But basically, in here we have again is titanium oxides. Um, we have different different plants. Well, not different plants. It's a plant which is exposed again to these materials, and we did some maps. We get several points, and I collected a database of about 40 or 50 spectra. And the main crystalline phases are again anatase and rutile. And my first idea was, okay, I'm not going to be looking at this spectrum by spectrum because it's going to take time. So I will compile this into a data table. Uh, sorry, at some point this gets a bit slow. There it is. So I created a data table with all the energy points. And basically I have region is from where it's coming from epidermis or from the cortex. Most of them are coming from the epidermis of the plant. In the class, I have like my treatment, so I know there's an 800 ppm of anatase, a biosolid, etc. And this is actually the part I want to test. I want to know if there's a difference between this and this and that. But in reality, when I look at the spectra like this, so now you see, you scratch your head like, what can you, other than finding this one, which is very, very different, and maybe this one, which is really in shape, is not so different from the other. It's hard to say like, okay, what do we have in there? And so you can probably use some of the classes that I created already. So for example, if I make the region, I see it doesn't really matter if it comes from epidermis. It doesn't help me see much, even if I show the averages. It's all mixed, right? So I couldn't see any pattern. Then I say, okay, let's try my treatments. It's the same situation. So then I said, okay, I'll try to investigate this with a principal component analysis. So I don't provide any supervision to the model. Before, I had to do some pre-processing. And the pre-processing basically in order to eliminate issues of this shape after. So I use an algorithm to convert this to second derivative where the maximum becomes minimum. And then I apply a normalization by vector because I'm not really interested in the changes in intensity. And then I cut some parts of the spectrum, and then this gets injected into the PCA algorithm. And I get this, shoot, I gave away my result. Basically, if I don't attribute any color, then, okay, I get here, I more or less see like there's a region there, a region there, a region here. I am expecting this to be a three-phase, three crystalline phases mixing. So that would be kind of like a triangle, right? So I introduced a rutile model compound, an anatase model compound, a rutile model compound, but I didn't have any other one. So somewhere in here, that is the pure rutile, and the pure anatase, and somewhere in here is the pure rutile. And then I selected this one, and I looked at the spectrum, and this gave me a good idea. Not this one, but this one. Sorry, because I have two screens and they appear in the other screen. When I clicked in here, I got this one. So now I said, okay, there is my three-phase, my triangle is here. And most of my data is, yeah, not very neatly inside, but it tells me something about it. So then I thought, okay, since I know the shapes of the spectrum, then I can maybe already pre-classify manually the data and attribute them to rutile anatase or this, which I call ilmenite. And I created those labels in the data table. And if I now use that as a way of shading or coloring or creating clusters, then you see there is this side, which is dominantly rutile, this part here, which is dominantly anatase. Then there is this one here, which I know for sure based on this very big intense peak, it's the ilmenite. And if I select this here, what is my problem here? Okay, I just have to well, I don't know, it's not working.
So while you want to do that thing, I noticed that, so this is the beauty of that part. So all that data, notice how the clusters got formed. And I think that's what Hiram wants to show us.
Yeah.
What that means, that outlier that you have there.
So I don't know why. Principle here, I recover. Yeah, there you go. So now I found this part here tells me the difference between rutile and anatase, also this, and then this pre-peak tells me the difference between ilmenite. And so now I am pretty convinced that the classification, which is manual, and because I only have 40 spectra, and I can do, it would be much nicer if I can do this in a more automated way, but it helped me understand the structure of the data. I know that these are not perfectly ilmenite. They're probably a mix of this and that. That's why they kind of have a tendency to go towards the anatase group. And that also makes sense. And at the end, so I recombined everything, and I got the averages. And what I managed to do is to decompose my 50 spectra into three main species, right? Again, it's the same as in the, because I had the experience with the sludge from Chihuahua, and it works beautiful. So it's the same case, right? You have some anatase because we are exposing to anatase nanoparticles. You have rutiles, and then you have this phase, which is the ilmenite, which is coming from soil, from the titanium in soil. And these are the averages. So each one is an average of about 15 or 16 spectrum. And I can show you, if you want to see how it looks if I shade it with, and of course there is overlap, right? But I know that there's a normalization issue, and that's a problem with my signal, but in the shapes, they are the same. And that's what I'm interested. So the shape of this minimum blue and the maximum, they're the same. Similarly in here, even though this seems to be a sharper peak and this was a bit more smooth, so that means that there is some of the other component in there. but I'm pretty happy with this. I think I cannot do much more on this data because the level of noise is not great. And so now I can, for example, this is my treatment anatase 800, and I can go and count how many times I found ilmenite and how many times I found anatase. And that's probably a good way of reporting the data. So I am trying to find to reach the final way of presenting the data. But okay, well, this is, I like here in Orange because I'm, like I said, I'm not a programmer. So I know that for you experts, you probably say, okay, I can do that in maybe 4 lines of code or something. But for me, this is very useful. I can, and also, let's say I am trying to get a bit better on working with code lines. But many of my collaborators don't know or don't want to do that. And the advantage of this is that I can save these and I can send it to my friend, and then he can link another data set and just adjust more or less my recipe. And it's very visual, right? And it's, I mean, it's so visual that you saw I put it on a slide, right? I could tell you in bullets what I did, normalization, the PCA, whatever. But in here you see it in this very visual way, and that's the part I like of this software. I think it's friendly. It's good to transfer and test different things, and we among our colleagues we share all these workflows. And at some point, then probably it's better if it's automated. Once you have something which is very well established, maybe you can convert it more into a kind of black box. I don't know. Okay,
Yeah.
so what I want to show, let's see. I
And
don't know if you
I'm
want
glad
to
that you show us this
or
part of Orange because Hiram just mentioned. I mean, I know a little bit of programming. I don't want to go more in depth on this part, but at the same time, you have the ability to incorporate your scripts here. Usually, I think you mentioned previously in our conversation that you can write a Python program or a
yeah,
Python script. You can just pull information from there without going into the details. And that is where the beauty. This is the frontier between
and
having
your
these scientists, this hardcore science that we have been working in many
here
years.
you can plug your Python script. So if at the
There is
end,
like there is the widgets, yeah. So this is one of the things that there is. This is the ability. This is the frontier, the borderline between computer scientists, programmers, information technologies, scientists. Here's where we put everything together. You can just drag and drop, like what you just said, some scripting. As long as you know how the data is and what is the output, this is very nice. I think we're running out of time. We have 12 more minutes. I have a question for you. It says, I have a question for Dr. Castillo. Once you recognize there is a negative impact in the environment, what happens next? Are there any regulations? That's one of the questions that I have from the chat. You want to elaborate a little
well,
bit on that?
one example I can give you. I mean, that's what we would like to have a contribution, and um, there are many examples of how we, from science like this, from research studies, experiments like this, we managed to put limits into how much you can put on a water stream or how much you can put into the air, etc. In the case of nanotechnologies, of nanomaterials, there are regulations. And so, for example, one example I have is about titanium oxide nanoparticles in food, in the food industry. This is, I'm not sure this is the case in the US, but in Europe, in the European Union, I think it's global now, or maybe it started with France and Germany, but there is this food additive E171. It's basically titanium oxide. Is not of nanoparticulate size, not all of it is nanoparticles, but a fraction of it, a percentage of it are nanoparticles. And based on research, and one of those studies is actually a synchrotron-based study, they demonstrated that if you keep ingesting titanium oxide nanoparticles, you can increase the chances of getting cancer of colon, because it goes in the gut and in the crypta of your intestines. They get accumulated, and then after this induces repetitive injury, right? Oxidative stress, and with time you can develop this. Now, it's not classified as if it is chromium 6, for example, or arsenic and other things that we know that are carcinogenic, like in the class A or class 1, I don't know which is this. But this is already classified to the point where in here in Europe, they ban this E171 food additive, which is you found, like I showed in my presentation, in baking products, in this like glassy sugar that you use, in this baking powder. You have it in chewing gum, this very colorful enamel cover coating that you have in the chewing gum usually contains titanium oxide. So, I mean, you add up, especially if you like candy, and candy and baking goods, like bread or things like that, donuts. You keep ingesting titanium oxide, and this causes some issues, right, for your digestive tract. So there is one example, but there should be many. I don't have right now more, right? But there is one example where, based on these investigations you do, you can say, if you get close, if your study is well done, and you go and communicate this to the regulators, then they should be able to create laws, right? And that's actually our objective, right? Not always we manage, right? Because sometimes our studies, they are not replicated enough, or they can have some issues with it. But we try to put the evidence out there, and then try to distribute it, and try to create conscience, and make us, at some point there should be some sort of regulation, right? I don't know any regulation, for example, for sunscreens that contain titanium oxide, but I know that personally I avoid them, the ones that have nanos. And it's not because they're going to cause any problem to my skin, because they would not, unless I have like some injury already. But I don't like the fact that I know that when I go in the lake, or when I go in the ocean, or when I go in the river, like if you go to a swimming pool, probably not a problem, right? But because there will be filters and whatever. But if you go to a natural stream of water, right, then it's going to wash off, and it's going to have some impact, especially because they will sediment, and they will be lying in the ground, and you have a lot of phytoplankton there, and small animals that are always living in the bottom of lakes and rivers and oceans and all this. So I know for sure that's not a good idea. So and in my personal opinion, so someone maybe argue with me, but this idea of putting nanoparticles is basically so that the sunscreen becomes more transparent. That's one of the reasons, right? It becomes more transparent, so when you put on your skin, then it doesn't look like you are all white from using sunscreen. It's kind of aesthetic application. There is some part of it which is not only aesthetic, but the main drive, I think, is, in my view, is aesthetic. So I don't mind. I can go away with the sunscreen that puts my face a bit white. I don't care. Like that, I know that it's still in there, right? So I try to avoid things that have nanomaterials in the sunscreens. But I don't think there is any regulation, not to my knowledge.
Yeah, and that's a great question, and I agree with that explanation as well. I think right now this is the interesting part about living in these days, when we can make, or we can recognize this analysis of having certain components with certain things. So now our question, is it possible to produce X-rays with cyclotron? Is there a difference between, is there a difference besides the size?
Produce X-rays with cyclotron, so not synchrotron, or what is? I didn't understand the question really.
I think it was meant of a synchrotron. Yeah. So the X-rays are a little bit different, right? Oh, so
Okay, okay, yeah. In terms of the kind of light, okay, yeah. The main difference is the intensity, and the intensity, it's the reason why they're so intense is because they are what we call collimated. They go in one direction, right? Whereas if you use another kind of X-ray tube, you're going to emit X-rays in all directions, and if you try to collimate this light to focus it, you're going to have difficulties, right? Because X-rays are difficult to focus. You have to use very specific kind of optics. It's not like optics for visible light. And so that's the advantage of the synchrotron. Like, if you see is the number of photons per millimeter square. If you don't have an area, and the probably in terms of number of photons, you're probably producing the same amount of an X-ray or of a synchrotron. But the advantage of the synchrotron is that they go in this very, very narrow cone, this very narrow, uh, our beam, our full beam is about 1.5 millimeter or something like this. And then from there, we use the special optics to focus it to nanometers, right? So we have a lot of photons in 1 millimeter. So it's this photon density, which is amazing in synchrotrons. But there are other properties. For example, the photons are polarized. And this is not a part that I explore in the techniques that I'm using, but there are techniques that are based on this thing of the light being polarized, because since you have electrons running and you have magnetic devices, so electric and magnetic fields, they will accumulate in one direction. So you have photons which are polarized. And there is another advantage also, because there are bunches, and the bunches, there is a time structure of the synchrotron. So if you want to do time-dependent studies, you have nanosecond distances between the electron bunches. So you have a nanosecond laser or a nanosecond light source that you can, but this is, I don't want to go too much into that because it's not what I do. So I don't want to make mistakes on saying too much, but the temporal structure and the polarization, these are also great advantages of synchrotron radiation that you don't get with normal X-ray sources. So it's the light the light density, the photon density, amount of light per unit area, the polarization, and the time structure. Those are the three main advantages of synchrotron.
Good. Thank you. Thank you for the question, and thank you for the. We have time for one more quick question, if it's possible. I think we still have in our range of time. Mm-hmm. So one of the things that I would like to thank you, Hiram, Dr. Castillo, who took the time. I don't know if you noticed, but the window that is in his lab, it has been changing through light, not too much light, and then now it's dark. The work that he has been done, you can just Google his name, right? There's a lot of publications in tier 1 journals and conferences and peer review conferences. And the work that he has done as an analyst in the synchrotron is outstanding. He, and of course, being my personal friend, one day was like, let's make this episode, because many of the things that you might be asking, okay, where's the programming, where's the algorithms, where's the complexity analysis and things like that. I want you to be aware that many of the problems that we're trying to solve in computer science, they're symmetrical to in other fields, like in the aspect that he's been working. We care about performance. We care about the impact. Yeah, you have the best and the worst case scenarios that you might have. Of course, we use different tools. We can use a different programming language in an operating system. They're using different mechanisms, different chemists, different chemistry, different composition of the aspects. They're very similar for what we have identified in computer science, computer engineering, even in terms of when we deal with reliable computing like cybersecurity and whatnot. There's always that component that we always gonna see symmetrical in other fields. It can be in chemistry, it can be in philosophy, it can be in software engineering, it can be in biology. So there's a lot of overlapping in different aspects. I think there's a lot of connection here of the work that we have done in computational sciences, in data sciences, that they intersect with the work that they've been doing. We need tools, we need experts, and of course, we need intersection, and these collaborations are very unique. I think one of the things that caught my attention since we started to have this conversation a couple of months ago, Hiram and I, is that we have in mind something about the ethical responsibilities of our work as a scientists, as an engineers, as a software engineers. and there's some impact on the ethics involved there. There's some implications, responsibilities that when we done certain products or certain research, there will be always some implications and responsibility. How will it gonna impact our environment, our society, and our future generations? And I think this is one of the main highlights that we want to obtain with this podcast, to identify these intersections and what people are doing in certain aspects in certain areas that will basically have this implications. I would like to thank Hiram Castillo for your time, for having this exciting discussion and talk about all your work that you share with us. Hiram, I'm very happy to have you here in the podcast and to make the time and effort and the research to come up with this cool presentation that we really enjoy. Unless there's some questions here in the chat or in someone who is being already present. Since it's not, or maybe, I
I
would like
want
to thank you,
to
Hiram.
just take a minute, or less than a minute, just to say thanks to you. And I really enjoyed all the discussions we had before the podcast. And I hope that we can continue to have every once in a while some nice chats, because I really enjoy that. But I want to say just thank you to like all the people that works at the Beamline, that worked before the Beamline, my colleagues from the other groups on ESRF, and all the institutes that collaborate. Like, I mean, it has been for me along my career very exciting to be surrounded with so many smart people, and I think that my contribution is somehow kind of small to all this like super cool work. So I just wanted to acknowledge that this is like a really big collaborative effort, and that I am not able to name all the people that is behind even the some examples that I have in here, but there are so many more that are a bit left out. And I hope that you liked the presentation and the discussion. And thanks to all of you and to Christian.
And we love it. And we were actually, now that you mentioned that, and thank you for sharing that, we would like to have, because there's so much stuff that we can just try to compile it in one episode, that we were thinking, Iram and I, to come up with a part number 2 when we can go more in details into other things. It doesn't have to be about nanoparticles or computer science. It can be about something else, because as I mentioned, we have a lot of things to do. Thank you, Iram, for your time. Thank you for sharing what the exciting work that you do in the synchrotroning with your team. I have noticed when every time I go into your papers, obviously you mentioned something, is a collaborate effort. So all the work that is being done in collaboration, I think that's a great team you guys have there. I think they're great scientists, great people. And thank you so much for that. You can read the messages that are in the chat. People are thank you very much, Dr. Castillo, for all the work that you have done. And thank you. I want to thank everybody who has, who attend to the podcast in live. Yeah. Thank you for your comments. Thank you for all of those who send the messages and the questions and their RSVP. We cover a couple of those questions that you mentioned. I just disseminated to him through the entire conversations that we had. And please join us for the next episodes. There's a couple of exciting things coming in this season of the podcast. We're going to have a more focus in artificial intelligence and of course in science as well. I think we have a little bit of cybersecurity involved, artificial intelligence, machine learning, and in the next episode. So I want you to please join the social media and the channel just to keep it posted. Also, please subscribe to the channel so you can get the information about the next episodes. Thank you very much for everybody to join us. See you next time.